Hi everyone, welcome to this live Q&A session following the fantastic presentation by Akoya Biosciences. It is my pleasure to welcome Virginie and Sebastian here for this Q&A session. And Sebastian, the first question we've received is for you. How could spatial data be used clinically?
Well, thank you for the question. I think we haven't had so far, let's say, any spatial metrics used in the clinics, but I can perceive that with the onset of becoming more popular, we could employ the distances in between cells as a predictive marker for diagnostics and or tied to as a therapy. So if there is a certain biomarker giving a spatial metric as being predictive for instance, IO checkpoint therapy success, there might be a good way of using these markers.
That's great. Thank you very much. Virginie, now over to you. Akoya offers two different platforms. What are the advantages of each platform and how are they complementing each other?
Yeah, that's a great question. And indeed, Gavin quickly mentioned that we have two different kind of platforms at Akoya. So the first one, called Codex, is really our discovery platform. So we can go up to 50 biomarkers using this platform. So it's a very discovery tool in the sense that it's made to discover novel biomarkers, predictive biomarkers, and really to get a deep understanding of what's going on, whatever the disease or tissue type we're analyzing. So in immunotherapy, for example, we see more and more publications where we use this multi-parametric platform to get a deeper understanding of what's going on in the tumor microenvironment at a single cell level. So really, this Codex platform is not only affordable and amenable to any lab because it can be plugged to any microscope, but it really enabled to get this complex analysis of what's going on. But once we have identified those very predictive biomarkers or clinically relevant biomarkers, we need to apply it to a bigger cohort. And so we need a higher throughput. And that's where Phenoptics and the Vectra Polaris comes into play. So the Vectra Polaris has been really developed to be more into a clinical tool. So now we have the ISO 13485 certification to respond to the GCP regulation. And it brings the high throughput that one needs to move into clinical studies and eventually into patient care. So we can scan 6-plex plus DAPI in less than 10 minutes. So it's a very fast imaging workflow.
That's great. Thank you very much. Right now, over to you, Sebastian. So the next question we received is the spatial data is not easy and quite complex to work with. In your opinion, what would be the major challenges and what would you advise for those who are looking into this right now?
That's a good question. So I think the biggest challenge actually is in repeatedly and precisely defining the cells, because the cells are everything will be traced back and computed back where a cell is located at and how good actually that cell is detected. And that in the end depends on very precise nuclear segmentation, because in most software packages, each detected nuclei will correspond to a cell, an event, and something like that. And this will then end up in as the origin or as the output for any spatial metrics, any spatial computation. So if there is no sufficiently good and precise nuclear segmentation, it will cause problems later on in any downstream spatial process. So, and any good advice would be just take a very good close look at how the nuclei are segmented.
Thank you great. Thank you very much. Virginie, now over to you. What are the benefits of single cell resolution and spatial phenotyping versus ROI and its promises in translational research?
Yeah, I mean, as we could see through this very nice spatial biology symposium, there are now lots of really fantastic multi-omics and genomics, spatial transcriptomics analysis, but it's all ROI based from what I could see and hear. And we believe at Akoya that every single cell, matters, and that we can really identify not only rare cells, but we also need to look at the exact distribution of those cells, and having a very good distribution analysis and being able to map those different cells thanks to X and Y coordinate, we can not only have a better understanding, but also start to predict response. I mean, as Sebastian showed during his talk, just looking at FOXP3 and B cells and CD8, you can already have increase a lot the correlation with the patient response to the drug you're studying. So yes, looking at single cell versus ROI is very important. And we have now some collaborators also looking at spatial, so transcriptomics on top of codex analysis, for example, to be able to map site-seq, and so gene sequencing back to the single cell analysis. And so really in that sense, you can have best of both worlds. So yeah, I think
that's great. Thank you very much. Now over to you, Sebastian. In which diseases might spatial data be useful for?
Well, I think most talks today and yesterday you heard have been dealing with immune checkpoint therapies in any malignant disease. But I can also, apart from, let's say, a disease that has a morphological correlate found in the tissue, like tumor cells or tumor islands and invaded immune cells, there might be other diseases that have any histological changes being observed in tissue. So there actually might be tuberculosis granulomas. There might be tertiary lymphoid structures in any other given diseases that see constant or enduring waves of inflammation, and these are then transferred into aggregates of immune cells that are found within the tissue, which probably have cells interacting with each other and exchanging signals based on receptors and ligands, and these ligands again then can be targeted by any kind of inhibitor or interfering molecule. So I would say mainly diseases where any change in tissue architecture and cellular distribution within the organ or tissue is obvious, and maybe even in diseases where we don't see any big changes in these markers based on H&E, we can probably or hopefully reveal cellular changes or cellular changes of activation markers based on multi-parametric profiling, like using the discussed platforms like Codex, Phenoptics, spatial transcriptomics, and henceforth.
Brilliant, thank you very much. And now the next question: what excites you in spatial biology, Sebastian? This is a question for you.
Okay. What I like, or what excites most, I would say, is first of all, is the beautiful pictures. It's not like looking on histograms or scatter plots from flow cytometry, but we actually see something, and it gets even nicer once we can trace that back to something happening in the patient, and then probably having groups of patients distributed differently, because some survive better, some have a more promising outcome. And if this can be then dated back to what we actually see on 1 or 2 slides and a couple of markers, that is highly fascinating, because there is so much information stored within the tissue, which can only be revealed if we use more markers, if we use or if we employ spatial metrics, if we employ spatial biology behind that. So there is actually a beauty within the numbers and a beauty within the colors, and that I find highly interesting and fascinating to work with.
That's great. Thank you very much. And this concludes the live Q&A session with Sebastian and Virginie. Thank you very much once again for joining us and also for the great presentation. If you have any questions, please feel free to reach out to them. I'm sure that they will be happy to have a chat with you. Thank you very much. Bye.
Thank you, Esther.
Thank you very much. Thank you, Esther.